This is a production of Cornell University.
My lab works on DNA methylation and epigenetic inheritance, and what I'll tell you about today is a problem that we've been working on for a few years, which is how do plants and other eukaryotes actually manage to inherit patterns of DNA methylation on chromatin. So how does epigenetic inheritance work around this structure where DNA is wrapped around histones? So I'll start the talk by introducing DNA methylation and epigenetic inheritance, and then I'll tell you about our work and trying to understand how this deals with chromatin. And then I'll finish up with some evolutionary implications of our work. So first, just to introduce epigenetic inheritance and how DNA methylation plays a role in this. So this is nicely illustrated by this Arabidopsis gene called FWA. And this gene is normally silenced in Arabidopsis plants in vegetative tissues. And it's silenced by DNA methylation that is present on the promoter and the start of transcription of this gene. And now, about 25 years ago, Martin Corneef's lab isolated a semi-dominant late flowering mutant that they called FWA. And when they tried to figure out what was causing this, they found this gene that was overexpressed in the leaves of these mutants. But there were no genetic differences between this FWA and this FWA. And the reason for this phenotype is that this FWA version is not methylated. So these plants are genetically identical. They don't have any DNA sequence differences. The only difference is that here this DNA is methylated and here it's not methylated. And these two methyl states these two states are very stable in the laboratory, at least. These plants will always be early flowering, and this gene will always be methylated and silenced. These genes will be always late flowering, and this gene will always be unmethylated and active. And in fact, they're stable enough that you can cross them together. So you can cross these plants with these plants, and these methylated and unmethylated alleles will stay methylated and unmethylated, and then they have they'll segregate as Mendelian alleles. And so this behaved as a perfectly normal Mendelian trait. And this isn't just a property that occurs in the laboratory. So around the same time, Rico Cohen's lab described a epigenetic allele in toad flax. So toad flax normally has bilaterally symmetric flowers. But a wild, normally occurring version of these species has these so-called peloric flowers, which have radial symmetry and very different arrangements of the flowers. And this is caused by methylation and silencing of the cyclodia transcription factor. This allele was actually described, as far as I know, for the first time by Linnaeus in 1750. So it's a very old allele, and it's been around for at least 250 years in natural populations of toad flax. So these methylated and unmethylated states can in nature be quite stable and persistent. So this ability of DNA methylation to carry epigenetic information has to do with how it's initiated and maintained. And what I'll tell you today is fairly specific for plants, but it works in a similar way in animals too. So this is really the plant-specific part, where in plants DNA methylation is initiated by this pathway called RNA-directed DNA methylation, which is a derivative of RNA interference. This is a very simplified, very simplified version of how this works. So a transcript is made that is recognized as aberrant in some way. Perhaps it's double-stranded. Perhaps there are other issues with this. And then it is converted upon transcription. And this transcription occurs by pol 2, but it also occurs by a plant-specific version of polymerase II called pol 4. and it's converted by an RNA-dependent RNA polymerase called RTR2 into double-stranded version that is then processed by a dicer nuclease into small RNAs. These small RNAs get loaded into a effector complex that contains an argonaute protein, and this appears to interact with a nascent RNA made by another derivative of Pol2 called Pol5 that has an accessory nucleosome remodeling protein called DRD1, and we'll talk about these nucleosome remodeling proteins more in a second. And this interaction somehow activates a DNA methyltransferase, which in Arabidopsis is called DRM2, that methylates the DNA, and it represses transcription from this locus. Now, the DRM methyltransferases and their animal homologs called DNMT3, they can, at least to some extent, methylate cytosines in any sequence context. So all the methylation I'll talk about today is methylation of cytosines, and this is the predominant form of DNA methylation in eukaryotes. But if in plants, if the RNA signal is turned off, then this methylation would disappear because after DNA replication, the old strand here in black keeps its methylation, but the new strand in red is synthesized unmethylated. So there's another methyltransferase system that is there to keep the methylation propagated across generations. And this is a methyltransferase called MET1 in plants and DNMT1 in animals. And what it does is with at least one accessory protein, it recognizes CG dinucleotides that are methylated on one strand but not on the other, and then it methylates the unmethylated cytosine. And because these sites are symmetrical, this catalysis essentially allows semi-conservative inheritance of DNA methylation patterns every time the DNA is replicated. And this is the core catalytic process that, as far as we know, makes DNA methylation epigenetic. So this allows for the methylation patterns to go through mitosis, and at least in plants, to go through meiosis very effectively. So in plants, there's yet another pathway for DNA methylation that I need to introduce. Hold on a second. I'll go on. I think I hope I didn't make a mistake. I might have copied the wrong version of my talk into this computer, but we'll see. If I did, we might need to do a little switch. So yes, because on that version of the talk, this slide was meant to come first. So I'll just go back. Right. So so I just first wanted to tell you about the function of DNA methylation in plants, and the major function appears to be to silence transposable elements. So the basic idea is that transposons will be methylated, and this methylation will be heritably maintained, and it will silence these transposons, and this will preserve the integrity of the genome. And then gene regulation of the kind I showed you with FWA will happen when a transposon will insert in the vicinity of a gene, and this will become methylated, and then this has the potential to silence the gene. There's also a type of DNA methylation in plants and many animals that's present within active genes, but we won't really talk about that. So today, the entire talk will be focused on transposon methylation. Now, I'll just go back. In plants, particularly in transposons, there's another DNA methylation system. So in addition to DNMT3 and DNMT1, there are chromomethylases, and these are methyltransferases, and the one in Arabidopsis is called CMT3, and that methylates a context called CHG. So it's called this because H stands for anything but G, and it's just done to separate this context from the one catalyzed by MET1. These methyltransferases are linked to a histone modification called H3K9Me2, which is the dimethylation of lysine 9 of histone H3. And so all kinds of DNA methylation, all these three pathways converge in transposons. And because these DRMs are not very sequence specific, their methylation is generally referred to as CHH, C anything but G, anything but G, to separate it from the kind that's catalyzed by MET1 and the kind that's catalyzed by CMT3. Oh, sorry. So I apologize for the slides mix-up. So the one final thing I wanted to mention is that it's well known in plants for quite a few years that pretty much all the CG methylation is catalyzed by this enzyme MET1, and all the CHG methylation is catalyzed by this enzyme CMT3. But not all the CHH methylation is catalyzed by the DRM pathway, right? So there was something else missing from this picture that was mediating some of this asymmetric CHH methylation. Okay. So now that I've introduced DNA methylation and epigenetic inheritance, kind of get to the heart of the talk, which is how this actually works within chromatin. Right? So everything I showed you so far is methyltransferase is methylating DNA, but this DNA is wrapped around the nucleosome, and this imposes certain limitations on DNA methylation. And these limitations, I think, became kind of clear quite a while ago when Eric Richards discovered this gene called DDM1. And DDM1 came out of a genetic screen for mutants with large losses of DNA methylation. And DDM1 turned out to be a type of protein called a nucleosome remodeler. That is basically a ATP-driven machine that does work on nucleosomes. So these are proteins that can assemble nucleosomes, they can move nucleosomes, they can space out nucleosomes, they can disassemble nucleosomes, they can take out a part of a nucleosome and put another part in. So they're kind of these work engines that work with chromatin. So the identification of this gene made it clear that the nucleosomes in chromatin had an important influence on DNA methylation. Later, people figured out that DDM1 loss of function causes major methylation losses in all sequence contexts. So it affects CG, CHG, and CHH methylation. It is required for methylation in transposons, but not in genes. So this gene methylation I mentioned is not affected really by DDM1. It has a close homologue in mammals called LSH that appears to do more or less the same thing. But how DDM1 actually interfaced with DNA methyltransferase systems, including RNA-directed DNA methylation, is really unclear. I don't know how it actually was mediating methylation in all the different sequence contexts. So initially, we approached this by analyzing DNA methylation genome-wide in DDM1 mutants and mutants that affected this RNA-directed DNA methylation. And the two mutants we worked with the most is RDR2, so this mutant in this RNA-dependent RNA polymerase, and DRD1, this nucleosome remodeler, which is specifically seems to be required for RNA-directed DNA methylation. It's a protein that's actually homologous to DDM1. And I'll be showing you quite a lot of these plots. So these are meta-analyses that show an averaging of all the DNA methylation of a particular kind of sequence. In this case, transposons. I'll always be showing you transposons. And transposons are aligned at the 5' end and the 3' end, and then the methylation across all transposons is averaged in both directions. So we're getting an average amount of DNA methylation for CG, CHG, and CHH. In wild type, in red, methylation is high for CG. Mutating RDDM doesn't have much of an effect on this. DDM1 has a very strong effect on CG methylation. And double mutants between DDM1 and RNA-dependent RNA polymerase, or this DRD1, so double mutants between DDM1 and RNA-directed DNA methylation are a little bit stronger, but just a little bit. But there's really very little methylation left in DDM1 already. For CHG methylation, the situation is a bit in between. The RDDM mutants have a bit of a stronger effect. DDM1 has intermediate levels of methylation, and the double mutants have very low levels. And for CHH, it's even more dramatic, where RDDM mutants or DDM1 have these intermediate effects, and almost all the methylation is gone in DDM1 RDDM doubles. So kind of the bottom line here is that DDM1 and RNA-directed DNA methylation are complementary methylation systems. And if you get rid of both of them, you basically get rid of almost all the transposon methylation in the Arabidopsis genome. And so the next thing we try to do is to link this phenomenon to the methyltransferases that we're doing it. And so the first two parts here are just what we already knew. For CG methylation, if we mutate MET1, there's almost nothing left. For CHG methylation, if we mutate CMT3, there's almost nothing left. And homologous CMT3 called CMT2 has a very small effect on this kind of methylation. But when we look at CHH methylation, we find that in DRM2 mutants, so this is the methyltransferase linked to RDDM, we have a partial loss of methylation, which looks exactly the same as the loss of methylation in RDDM mutants. But in the CMT2 methyltransferase, this chromomethylase that is homologous to CMT3, there's a very, very strong loss of methylation. And between these two, they account for all the non-CG methylation, all the CHH methylation in the genome. So CMT2 was this missing bit that was responsible for DNA methylation in Arabidopsis. And I think you probably are noticing that the shapes of these lines are different, and the mutants that belong to the same pathway resemble each other. So RDDM mutants have this kind of bell-shaped shape, and DDM1 mutants or CMT2 mutants, they have this smiley face shape. And this is actually quite informative about what they're doing. So this is just a zoom in on the CHH methylation plots. So what we're doing here is we're aligning transposons from either edges, and then we're going into the middle. So close to the points of alignment, the methylation average is either small transposons or the edges of longer transposons. So if we move in further and further in, we're getting methylation from bigger and bigger elements, right? So by the time we're in here, it's 4 kb in, right? So only large elements will be averaged here. And so these different shapes actually tell you about the kinds of transposons that are being methylated. So in RDDM mutants, it's the smaller transposons that are here at the edges that are losing methylation. And in CMT2 and in DDM1, it's mostly the larger transposons that are here in the middle. And this is a more formal analysis of this, where here we're averaging methylation with respect to the size of transposons. And in wild type, there's not much of a relationship. But in DDM1 and in CMT2, the bigger the transposon, the bigger the loss of DNA methylation. And for RDDM, for DRM2, or for DRD1, the bigger the transposon, the smaller the loss in the DNA methylation. Right? So different sizes of transposable elements are relying more on different systems. RDDM is more for smaller elements, and DDM1 and CMT2 for this kind of methylation are more for larger elements. Which is interesting, but it's not terribly satisfying. Why does transposon size matter? And actually, what matters isn't the size. What matters is the type of chromatin these transposons inhabit. It just so happens that smaller transposons in the Arabidopsis are more euchromatic, and bigger transposons are more heterochromatic, and that's actually what makes the difference. So this, I'll show you that just in a bit. This is just to help you visualize what's going on in a particular example. So this would be a short transposon in the Arabidopsis. This would be a long one. And this is the methylation in wild type. And for the short transposon in DRD1 and DRM, all the methylation is gone, but in DDM1 and CMT2, it's still there. For the long transposon, in DRD1 and DRM2, the methylation is mostly there, except right at the edges of the transposon. And for DDM1 and CMT2, it's the opposite. It's the methylation at the edges that are still there. But the middle is gone. And in the double mutants, all the methylation basically is gone. And these remnants of methylation here correlate with the small RNAs. So this pathway is driven by small RNAs, and so in the absence of DDM1 and CMT2, the little bits of remaining methylation are catalyzed by RDDM through the small RNAs. Okay. So this is now an analysis that looks explicitly at this question of chromatin. And so for this, we're confining ourselves for smaller transposons which are smaller than 500 base pairs, so that size variation is not an issue. And we're just here calculating correlation coefficients. So each of these bar shows a correlation coefficient between the loss of DNA methylation in DDM1 in 3 different sequence contexts, so CG, CHG, CHH, associated with a particular feature of chromatin. And what this shows is that nucleosome densities and histone modifications associated with heterochromatin, like HGK9 dimethylation, are positively correlated with loss of methylation in DDM1 mutants. And histone modifications associated with euchromatin are negatively correlated. So a DDM1 is much more required for heterochromatic transposons, and it's much less required for more euchromatic transposons. And the same thing in reverse is true for methylation guided by small RNAs. So this is the same kind of analysis for methylation that depends on DRD1. It's the component of RDDM. And here, nucleosome density and heterochromatin are negatively correlated, and euchromatin is positively correlated. So RDDM doesn't work very well in heterochromatin. It works much better in euchromatic transposons. But there's one feature here, really, which isn't heterochromatin or euchromatin, it's nucleosomes. So nucleosome density correlates negatively with RDDM and positively with DDM1, and we'll get back to that a little bit later in the talk. But basically, for now, heterochromatin DDM1 and euchromatin RDDM. So this is just a model summarizing this part of the talk, where we have a typical long heterochromatic transposon and a short euchromatic transposon. And they're methylated differently. The euchromatic transposon will be methylated by this RDDM system, and the CHH methylation will be mostly guided by DRMs, whereas the heterochromatin requires DDM1 for DNA methylation, and this somehow allows methylation by MET1 and CMT3 and CMT2 of this transposable element. So this links DDM1 to heterochromatin, but it doesn't really explain what it is about heterochromatin that makes DDM1 important. Why is it that we need DDM1 heterochromatin more than a euchromatin? And this is where our work with DDM1 intersected with our work with another component of chromatin, linker histone H1. So H1 is a very abundant protein that's found in most eukaryotes, including in plants and in animals. And this is a highly schematic picture of this. So this is a nucleosome. This is DNA coming in and out of the nucleosome. H1 binds to the DNA between nucleosomes, the so-called linker DNA. This is why it's called a linker histone. And it also binds, though, to the nucleosome. And this dual interaction with the linker DNA and the nucleosome is hypothesized to kind of be a vice that stabilizes the nucleosome. And so H1 is associated with heterochromatin. It's associated with inaccessible chromatin. It can impede nucleosome remodeling. And we became interested in H1 because it's been reported to cause interesting and strange effects on DNA methylation in various species. So H1 mutations in the fungus called Ascombylus were shown to cause overall increase in DNA methylation. Basically, across the genome, methylation would increase. In mice, H1 mutations were published to have a localized losses of DNA methylation. So certain particular regions of the genome would lose DNA methylation. And Arabidopsis there's a study suggested that there were sort of random effects in DNA methylation. H1 mutations would cause increases and decreases more or less randomly throughout the genome. So, very curious about this. The first thing we actually did is to map H1 across the genome, just to see where it goes, and not surprisingly, it goes to heterochromatin. So there are two H1 genes in Arabidopsis, H1.1, H1.2, these are ChIP-seq data where both of them localize to around the centromere, and this is where heterochromatin is. This is where most transposons in Arabidopsis live. So this protein is highly enriched in heterochromatin, and we already sort of knew this from cytology down many years ago. This is a enrichment of H1 in transposons, because transposons live in heterochromatin. If something is enriched in heterochromatin, it will generally be found in transposons. Both versions of H1 are highly enriched in transposons, but they're specifically enriched in heterochromatic transposons. So if we separate based on histone modifications, transposons into heterochromatic and euchromatic, then heterochromatic transposons are highly enriched for H1, and euchromatic transposons are not enriched at all. H1 is in Arabidopsis a heterochromatic protein. It's not only in heterochromatin, it's actually found in genes too, but it's highly enriched in heterochromatic transposons. And then when we looked at DNA methylation, we saw something that was quite interesting, that H1 had kind of a dual effect on DNA methylation. Its most obvious effect across the genome was an increase. So in all three sequence contexts, CG, CHG, and CHH, H1 mutants had more methylation in transposons than wild type. And this was particularly dramatic at CHG methylation, but it was in all three contexts. But the shape of this methylation was different, right? So it's a lot, this curve is a lot steeper here than the wild type. And for CHH methylation, you can actually see it intersects. And what it basically means is that shorter, more euchromatic elements are actually losing methylation, and longer, more heterochromatic elements are gaining it. And this is a more formal analysis of this, where we are dividing transposons into heterochromatic and euchromatic types. And then we're comparing methylation between H1 and wild type and plotting the overall distribution of the differences between the H1 mutant and the wild type. So these curves here represent the distribution of the methylation differences. And so if the curve is shifted to the right of this dotted line, it means it's more methylated in H1. If the curve is shifted to the left, it means that these sequences are less methylated in H1 mutants. And so for all three sequence contexts, heterochromatic transposons are hypermethylated in H1 mutants. And they're hypermethylated pretty evenly. So these brownish curves have a kind of nice Gaussian shape. Euchromatic elements are shown in red, lose DNA methylation in H1. And they lose it a bit less evenly, especially for CG methylation. There's this long tail, and so there are some sequences which have very strong loss of CG methylation in H1 mutants. And so I think this helps to reconcile some of the conflicting DNA methylation data that have been published about H1. So it does have two effects. It causes losses of methylation and gains, but in different sequences. It causes increases in heterochromatin and losses in euchromatin. And this generalized increase of DNA methylation in heterochromatin looked a lot to us like the opposite of what happens in DDM1 mutants. In DDM1 mutants, we have a generalized loss of DNA methylation in heterochromatin, and here we have a generalized gain. So we're curious what would happen if we actually put these mutants together. And what happens is that in the absence of H1, the DDM1 phenotype is greatly relieved. Okay. These are these same kinds of plots shown for H1 now in green, wild type in red, DDM1 in blue, and two replicates of compound mutants between H1 and DDM1 in brown. And in all sequence contexts, the amounts of DNA methylation in these H1 DDM1 compound mutants are much higher than DDM1. So And this is particularly dramatic for CHH methylation, where here basically the compound mutants look like H1. And this is just examples of what this looks like at heterochromatic elements, where wild type is in red, H1 is in green. And I think you can kind of see, if you look closely enough in this element, this effect where methylation is increased in H1, that this very jaggy kind of outline turns out very smooth. And in DDM1, these transposons lose methylation, as pretty much all of them do, and in the compound mutants, this loss of methylation is largely reversed. And if we examine what happens in these compound mutants carefully, what we actually find is that this methylation dichotomy between euchromatic and heterochromatic transposons that exists in DDM1 mutants disappears. So now heterochromatic and euchromatic transposons are behaving more or less the same with respect to methylation loss and DDM1. So I'll walk you through this analysis. These plots, each of these lines represents the ratio of methylation between a mutant and wild type. So H1 is a ratio of H1 to wild type. This blue line is the H1 DDM1 compound mutant versus wild type, and the black is DDM1 mutant versus wild type for the 3 sequence context. So for DDM1, it shows the characteristic DDM1 phenotype where, and then we are arranging them with respect to heterochromatin. So the most euchromatic transposons are here, and the most heterochromatic transposons are here. And this is counted as the amount of this histone modification. So transposons are arranged from euchromatic to heterochromatic. And as we get from more euchromatic to more heterochromatic transposons, the DNA methylation drops in DDM1 mutants in all 3 sequence contexts. And in H1, these red lines, you see the opposite. So there's a bit of loss in euchromatic elements and a bit of gain in heterochromatic elements. And this occurs in CHG and this occurs in CHH. When we look at H1 DDM1, what we find is something that looks very much like H1. Right? There's a relatively flat relationship for CG. There's the same kind of shape here as for H1 for CHG, and almost identical methylation in CHH. And so if we actually take H1 out of the equation, if we just superimpose these patterns, so we sort of treat H1 as the background and compare H1 to H1 DDM1, then we get relatively flat lines for both of them, for all the contexts. So what this is saying is that when H1 is gone, then euchromatic transposons and heterochromatic transposons lose DNA methylation to about the same extent in DDM1 mutants. And so now the more detailed model is that at least a major thing that makes heterochromatin require DDM1 is H1. Right? That it's the presence of H1 in heterochromatin that really requires DDM1 activity to allow these methyltransferases to do their jobs. But this model, though, still doesn't explain what it is that DDM1 does exactly. What kind of activity does it perform that allows methyltransferases to do their jobs, and how does it actually interact with H1? Does it remove H1? Is that what it does? And if that's what it does, why do H1 mutants have only a partial rescue of the DDM1 phenotype? So we did a number of things to address this. And one of them is to examine the relationship between DDM1 and H1 DDM1 and DNA methylation and nucleosomes. Since DDM1 is a nucleosome remodeler and supposed to work on nucleosomes. So before I get to the data, I just want to conceptually explain what we are up against. In complex genomes like Arabidopsis or humans or mouse, nucleosomes are not arranged the same way all across the genome, and they're not arranged with equal reliability either. So there's some places in the genome where nucleosomes are positioned reliably, which means that if you look at chromosome one chromosome, another chromosome, a third chromosome, 10,000 chromosomes, the nucleosomes are going to be all pretty much in the same place. And when you average them together with whatever analysis you're doing, you're going to get a nice representation of the nucleosome placement. But in other parts of the genome, there are nucleosomes that are positioned poorly. So in one chromosome, nucleosome will be in this position, and another chromosome will be in this position, another chromosome will be in this position. This doesn't mean these nucleosomes are not stable. It's just that every time chromatin is assembled after cell division, the nucleosome will occupy a slightly different place. And when you average all of this, you get something like that, right? You don't really get much useful information out of this. So if you're trying to understand how something like DNA methylation relates to nucleosomes, the first order of business is to find places that look like this, because there is where you can learn something. Places that look like this basically scramble all the information about where nucleosomes really sit in each individual piece of DNA. So with this in mind, I'm going to skip a lot of analysis and kind of just get to the point. This is a heat map where the redder the color, the higher the feature. And so this is a heat map of nucleosomes in H1 DDM1 mutants. So each line is a different piece of DNA, and this is centered on a well-positioned nucleosome. And so these represent these reliably positioned areas of the genome. These are places in the genome where nucleosomes sit in more or less the same places in all the cells. Okay. And then if we look at DNA methylation there, what you get is something that looks similarly equally well arranged. So this is now CG methylation, these H1 DDM1 mutants. And so red will be more methylation, blue will be less. So methylation is in these well-defined stripes that are in between the nucleosomes. And this is just a averaging of this type of data compressed into a single single averaged graph. So the gray are the nucleosomes. So this would be this bright nucleosome here. And the solid lines are DNA methylation in the CG context. The blue is H1, black is wild type, brown is H1 DDM1, and orange is DDM1. So this brown line just represents the average of this data, where DNA methylation is now highly periodic, and it oscillates between very low levels of DNA methylation in the nucleosome cores, where DNA is wrapped around the histones, and nearly wild-type levels of methylation in the linker DNA between nucleosomes. And you can see the same thing in to the much smaller extent in DDM1 mutants, where there's more methylation remaining in the space between nucleosomes, and there's essentially no methylation where the nucleosome sits. And see the same thing for the other kinds of DNA methylation. This is for CHG. Now you can see that this oscillation actually exists in all genotypes, including in wild type. There's a preference for nucleosome linkers, but it's much more dramatic in H1 DDM1. And it's the same for CHH. I didn't put H1 and wild type here because the lines intersect and it'll be much too confusing. But it basically looks similar to this, where you have this periodicity that is very prominent in H1 DDM1 double mutants, but not in DDM1. Now let's see if okay, good. Remember I told you that DDM1 actually has a mammalian homologue called LSH. And so we wanted to see if we could basically see the same kind of phenomenon going on in LSH mutants. And there's DNA methylation data available for LSH mutant mouse cells. So this is a graph where what we're doing is we're aligning sequences centered on the binding sites of a protein called CTCF. And CTCF is a mammalian protein that is well known to orient nucleosomes around it. So it kind of forms well-positioned nucleosomes arrays around it. And it also depletes DNA methylation where it binds. The gray line here are nucleosomes in mouse cells around CTCF sites, and they show some periodicity. And the blue line is wild-type DNA methylation. The red line is LSH mutant DNA methylation. And in wild-type, there's a little bit of periodicity here, but in LSH, there's much more pronounced periodicity. It looks a fair bit like the periodicity in DDM1. But this analysis is much, much cruder than what we did here in Arabidopsis. Now, this is actually something that my postdoc just did yesterday. So this needs a lot of work, but I just wanted to show this to you because I think it shows that this really works more or less the same probably in plants and in animals. And I'll try to explain what we think these data means using this model. At least a large part of how we think this works is that if we start with chromatin, the DNA is wrapped in nucleosomes, and in between it's associated with linker-histone H1. And then what DDM1 does is it does what nucleosome remodelers do. It moves or partially disassembles nucleosomes, or perhaps what it can do is can make a loop of DNA that comes out of the nucleosome. Through this activity, it makes the DNA accessible to methyltransferases like MET1. Because H1 actually doesn't just bind the linker DNA, it also binds to the nucleosome surface. It's very hard to imagine how DDM1 can do what it does without also disrupting contacts with H1. So we think it comes in there, it remodels the nucleosomes, it kicks out H1, and it exposes DNA to the DNA methyltransferases. If we don't have H1 in this system, then the linkers are that much more accessible to DNA methyltransferases. And this, we think, is a part of the reason why in H1 mutants, heterochromatin is hypermethylated. Right? Because linker DNA is more methylated. And you can see this here, where in H1 mutants, linker DNA is more methylated. It's actually true for all sequence contexts, including CGs. Just you can't really see the oscillation very well at this scale. When you don't have DDM1, then there isn't a remodeler to allow the methyltransferases to access the DNA, but the H1 is not as stable as the nucleosomes. It's known that H1 comes on and off. It's a relatively stable protein in its association with DNA, but not nearly as stable as nucleosomes. And so this dynamic binding and unbinding of H1 allows some access to DNA methyltransferases in DDM1 mutants. And so this, we think, is why you see some methylation in the linkers of H1, of DDM1 mutants, but not in the cores. And then in the double mutants in H1 and DDM1, there still isn't a remodeler to allow access to the nucleosome cores, but the linkers are relatively available because H1 isn't there. And because H1 is concentrated in heterochromatin, this removal of H1 has a much stronger effect on heterochromatin, right? You lose H1, and heterochromatin becomes much more accessible, and euchromatin doesn't change too much. And so then in H1 DDM1, we get this really big difference in heterochromatin between cores and linkers, because the linkers are accessible now without H1, but the cores are still relatively inaccessible because DDM1 isn't there. Okay, so that's the basic model how well we have it developed so far. It's not quite this simple, but this is the first approximation of how we think H1 DDM1 influence DNA methylation. And so just to finish up the talk, I want to go a little bit into evolutionary biology. Because in my lab, this is actually a substantial interest, try to understand how DNA methylation evolves. And we've profiled DNA methylation in a number of different species. And when we saw this pattern of methylation, this was a pretty strong feeling of deja vu, because we've seen this before. And we've seen this before in other species. So in our analysis, some of the species that we've looked at is a particular group of green algae called the Mammillaries. Green algae, which is to say plants, are a very diverse group of organisms, which embryophytes, which of which length plants are part, make up just one branch. And our knowledge about these guys in general and about the DNA methylation specifically is highly limited. So we have a lot of knowledge about Arabidopsis DNA methylation is present in these sequence contexts. We have some knowledge about DNA methylation in a couple of other groups of algae, like the group Berkeley and also the species called chlorella. But a lot of these other groups here are a bit of a mystery. And so we looked in this particular group, which we did partly because they're very, very tiny. These are the smallest free living eukaryotes. And we were pretty curious to know what they were doing with their DNA methylation. And both of these guys, both of these species that we looked at, have CG methylation in their genomes. And these species are actually quite diverse. They're about 500 million years diverged from each other. But the pattern of methylation was very odd. This pattern of methylation was, these are representative parts of the genome from these two species, where the methylation is in blue, and the exons are in green. So these species have methylation throughout their genome, except in promoters. And it's present in this very regular pattern, where there's a short patch of DNA methylation followed by a gap, and a short patch and a gap, and a short patch and a gap. And this pattern is nucleosomal. So by mapping nucleosomes, what we could show is that the methylation fits between the nucleosomes. So in these species, this is now representative genes. Methylation is in blue, nucleosomes are in gray, and the methylation fits right between the nucleosomes. This is a more general analysis where we're aligning at 0 the first methylated base in each one of these methylated clusters. So we're taking, for our starting point, the transition between unmethylated and methylated DNA. And averaging methylation going out 500 base pairs in both directions, or nucleosomes in gray. And so what these analyses show is that actually the relationship between DNA methylation and nucleosomes is very, very strict in these genomes. So the methylation is all present between nucleosomes, and it's basically never present in the nucleosome core. Right? So these genomes have everywhere, and by default, the kind of methylation pattern that we find in Arabidopsis transposons when we mutate H1 and DDM1 together. And the thing is, this kind of methylation pattern wasn't just present in these green algae. It's actually present in several very deeply branching lineages of eukaryotes. So this is a reasonably representative tree of eukaryotic diversity, where the groups that have this kind of linker-specific DNA methylation are in red, and other groups that have DNA methylation everywhere are in green. What was very puzzling about this was that this linker-specific pattern of methylation was interspersed with species which didn't have it. Right? It wasn't confined to one particular part of the tree. So this group of green algae I just told you about has this pattern of methylation, but other green algae don't, and land plants don't. Haptophytes have a very, very similar pattern DNA methylation to these guys. They, however, are very distantly related to other eukaryotes, so it's hard to make much of these. But in straminophiles, brown algae have a pattern basically indistinguishable from these green algae, but diatoms, their relatives, have a very normal-looking methylation of transposons that doesn't appear to be confined to linkers at all, at least based on looking at the whole genome. None of the animals or plants or fungi we've looked at has very highly linker-limited methylation either. So we couldn't really understand how this was possible, but I think our data with H1 and DDM1 suggest an explanation. It's actually not that difficult to transition between these two kinds of patterns. We can convert Arabidopsis, at least in part of its genome, from a overall methylation pattern to a linker-specific pattern by making basically 3 mutations, DDM1 and 2 H1 proteins. So I think it's relatively easy to imagine how small regulatory changes in nucleosome remodeling can give you a species where methylation is only in linkers or where methylation is everywhere. So I will end here, and I'll just thank the people who did the work. The CMT2 analysis was done by a graduate student, Ping-Hang Zi. The early work on H1 DDM1 was done by a former postdoc, Asaf Zemak, and a former graduate student, Yvonne Kim. The more recent work on DDM1 and LSH was from a current postdoctoral fellow, David Lyons, and the evolutionary work I mentioned at the end was that of a former postdoc, Jason Huff. So I'm done here and happy to take any questions.
This has been a production of Cornell University, on the web at cornell.edu.